So the paper this time is Synaptic Architecture of a Memory Engram in the Mouse Hippocampus by Uytiepo et al. And let me see, I gotta be able to move the slides, of course. So the study here was motivated by findings about drift of cellular representations over time. At least that's the impression I'm getting from reading the paper. And we discussed one possible example of something like this in a recent journal club on April 22nd, when we went over the paper on reconstructing a new hippocampal engram for systems reconsolidation and remote memory updating by Lay et al. That paper also examined hippocampus, though it focused mainly on representations in dentate gyrus and change or drift in the representations that activated or were recruited during later reactivation and updating of contextual episodic memory. So this may be a somewhat different case of drift or change than what motivated this study. In this study, EM reconstruction is able to give insights that light microscopy simply doesn't have the resolution to be able to do. This was another point they made in why they chose this method and why this particular study. The study wanted to test whether synaptic sites on Schaefer collateral fibers between CA3 and CA1 show evidence of Hebbian learning or not, where Hebbian learning, or maybe STDP if you want, means correlational or causational strengthening of connections based on synchronous or near synchronous pre and post synaptic activity. It's not entirely clear to me whether they started out suspecting non-Hebbian mechanisms in their involvement in that kind of drift, or whether they started out interested in representational drift and the data led them to focus on non-Hebbian indicators. There has been evidence of non-Hebbian components in learning or memory recall in psychology, cognitive science, and neuroscience. For example, recent research suggests that priming might be mediated by a complex interplay of mechanisms, including synaptic scaling, which is a form of non-Hebbian plasticity that adjusts overall synaptic strength to maintain network stability. In any case, they list a number of papers providing both support for Hebbian learning as well as some alternatives. And one of the papers they listed is an interesting read. For example, Mingi and Greenberger from 2020. This is one that contrasts several biologically plausible learning methods and includes, for example, behavioral timescale synaptic plasticity as a non-Hebbian one-shot learning that requires no postsynaptic spiking. That particular method utilizes an excitation-inhibition balance and a calcium plateau on apical dendrites that potentiates arriving excitatory inputs. In a sense, this reminds me of ADP-driven sustained firing at pyramidal neurons, though it's not the same mechanism exactly. Methods used in this study are quite relevant to our interests in methods that can work with static snapshots, as they use serial block phase scanning electron microscopy, or SBEM. They were able to do this for multiple specimens, so multiple mice, some of which were exposed to contextual fear conditioning, while others were exposed only to the context stimulus. They combined behavior, chemogenetic tagging, and detailed morphological analysis. Well, it says main findings here, but actually the next slide also has main findings on it. It was just too much to put on one slide. These are the main findings after one week after exposure, then doing the study, basically. They found evidence by patch clamp of increased firing thresholds on what they're calling PN+, that's pyramidal neurons that activated during the training, and therefore that were labeled. We'll get into the labeling in a bit, but no widespread significant increase of excitatory synaptic strength. Spatial distribution and total spine counts on PN+ and PN- dendrites were indistinguishable after EM reconstruction. Presynaptic terminal counts and distribution were also indistinguishable. What they did find were elevated levels of multisynaptic boutons on PN+ axons and a corresponding relative loss of single synaptic boutons. There were also proportionally more MSBs, multisynaptic boutons, that contacted more than two spines. They found that this amounted to a stimulus-specific reorganization of presynaptic axonal network connections. So continuing the findings, and yet the MSB, so multisynaptic Bhutan target selection, did not appear to be biased towards postsynaptic or PN plus, postsynaptic PN plus, in other words, co-activating cells. It didn't seem to depend on that. Their interpretation is that memory encoding involves local input-specific restructuring of synapses. They base this not just on the lack of significant postsynaptic modification, but also on the recruitment of PN minus postsynaptic neurons via presynaptic restructuring. The spines of neurons contacted by presynaptic PN plus axons were enlarged for both spines on single synaptic Bhutans and multisynaptic Bhutans, and the presynaptic terminals were enlarged, again, for both single synaptic Bhutans and multisynaptic Bhutans. In short, there was evidence of contextual fear learning related synaptic change, but it appeared to be presynaptic activity dependent rather than clearly Hebbian, did not appear to require postsynaptic activation. One could imagine that this is helpful, for example, for the recruitment of CA1 cells into an activation path. Presynaptic activation was also correlated positively with anchoring of mitochondria, especially at multisynaptic Bhutans. They then go on to argue that Hebbian plasticity does not account for the observed rewiring of cellular substrates underpinning memory traces. This may be controversial and may go a bit far given the temporal characteristics of the study, which is something I'll get into later towards the end. Finally, they go on to make some hypotheses about a relationship between these processes and age-related memory deficits. That connection was not clear to me and seemed very speculative, and maybe it was an obligatory sentence for the connection with their NIH funding source. That's just me hypothesizing. All right, so first of all, let's talk a bit about the workflow, and this is their labeling workflow. For image sampling from multiple animals, they focused on the stratum radiatum area of Schaefer collaterals into CA1, and they devised a workflow for irreversible labeling of transiently activated populations with a genetically encoded engineered peroxidase, Apex2 MGFP. This plasma membrane anchored EM-compatible marker was introduced with the Cre recombinase inducible adeno-associated virus, AAVDJ-DIO Apex2 MGFP, which I am not going to repeat that whole thing, into the hippocampus of previously characterized knock-in mice that express destabilized Cre, or DD Cre, under the control of endogenous FOS promoter. FOS is an immediate early gene whose transcription is restricted to a narrow window following neural excitation. Now, the decay of the DD Cre can be blocked by peripheral delivery of blood-brain barrier permeable antibiotic TMP. So the virus was injected into CA3 and CA1 of FOS-DD Cre mice to label co-activated glutamatergic pyramidal neurons in both areas. And then TMP was acutely administered 30 minutes post-training to allow the DD Cre transcription and translation. The expression of Apex2 MGFP was only observed after TMP delivery, and it was strongly upregulated by contextual fear learning, 4.5-fold. And then I'm just going to finish the slide before I take the question. So whole cell current clamp showed elevated firing thresholds and no widespread changes of excitatory synaptic strength in CA1 PN plus cells. And let me see who is.
It's me. I'm just wondering if you, or I guess Ken, knows what exactly is it that Apex2 MGFP does to be become like labeled on EM specifically. Like, I don't really understand the basic mechanics, and it didn't really describe it in the paper, and I guess I was a little bit lazy and couldn't easily find on a quick Google. Do you know how it specifically gets labeled?
So let's open that question up to someone who knows more on the call.
Okay, I'm going to go out on a limb unless there's somebody that knows more. I believe this is a singlet oxygen generator that is used in kind of a DAB reaction. Don't trust me on that, but I believe that is the mechanism.
Thanks, Ken.
Okay. Was that it for this slide? Or were there more questions about the labeling method or the protocol? All right, I'll carry on. Okay, the next part of the workflow was their reconstruction workflow. They sampled about 100,000 cubic millimeters EM stacks from CA1SR to ensure that the fear-conditioned mice had retrievable memory 7 days later at the time of tissue collection for EM. The behavior of separate cohorts from both groups was assessed upon their return to the original context. 3D EM stacks were aligned from raster images with 4 nanometer pixels, 2 microseconds dwell time, and 60 nanometer Z steps. And each stack contained approximately 200,000 synapses. Automated segmentation was used for the accurate prediction of plasma membranes, axonal and dendritic shafts, nerve terminals, dendritic spines, postsynaptic densities, and intracellular organelles. And this was benchmarked against manual reconstructions. The excitatory circuits were then reconstructed from the segmented images. Also, this image you're seeing in K here, that's about 1/12 of the typical stack size. But, oh, I should go back then. Yeah, go ahead.
Yeah, just a quick question. So how many different mice did they do in the control and the CFC conditions for this EM study?
This is terrible. I had told myself I was going to get that and make sure I knew that number, and then I forgot to do it as I was working my way through. Did anyone else pick up on the number of mice involved in the study?
I remember it being, I think it's N of 4 or 6. I'm seeing N of 4 in one of the figures. Yeah, something like that. I'll hunt it down and put it in the text.
So would that be 2 for CFC and 2 for CS, or do you know how they divided that up?
Yeah. Sorry, I'm hunting it down. I'll put it in the text when I find it.
Okay, yeah, if you find it, please put it in there. Yeah. Actually, I can try to find it myself as well. I've got the text right here. It should be in the supplemental materials, if not in the main text.
Well, I will say it's very impressive that they did multiple of these EM volumes. The EM volumes might not be that large, but the fact that there's multiple animals in this is kind of impressive.
Right. Yeah. Gosh, why is that hard to find? Wow.
I can withdraw the question.
We can come back to it maybe at the end. For some reason, it's not an immediately obvious number. Or we'll just go with the 4. We can do that too. All right, let's carry on for now. Okay, so now I'm going to work my way through the experiments one at a time as they work their way towards, I guess, their overall answer. The first one is looking at synaptic site distributions in CFC versus CS. They mapped dendritic spines of CA1 pyramidal neurons, postsynaptic compartments that cluster receptors and other signaling molecules near the sites of transmitter release from opposing axonal terminals. And they found that spatial distributions and total spine counts on the dendritic arbors of minus and plus neurons were virtually indistinguishable. They mapped the synaptic presynaptic terminals along the Schaefer collateral axonal fibers coming from CA3, and there they found the distributions and total terminal counts on minus and plus axons were nearly identical, as shown in that E graph here. It's almost the same curve. To determine if task-specific coactivation of CA3 and CA1 pyramidal neurons is attributed to their preference for wiring with each other, they compared the patterns of reporter expression in Schaefer collateral fibers and their postsynaptic partners within local receptive fields, and found percentages of plus spines receiving inputs from minus and plus axons were not significantly different. I didn't include that figure because I wanted to make this a little easier to read than the last time I presented with so many figures. The next thing they did is to look at multisynaptic Bhutan and single synaptic Bhutan proportions, again in CFC versus CS. Yep, there's a question.
I'm sorry, Randall. I'm being dumb today. Could you go back to the previous slide? And because that is a really important slide, right?
It is,
or I just want to make sure. So, and I was having a hard time digesting that when I was reading through the paper. So the curves in C and E are really saying that there's not that more synapses are not being created between the plus and plus, right? The
right
engram axons and the engram dendrites in CA1. Is that correct?
That's how I read it as well. Yeah.
Okay. And there
in spine count and distribution and no change in terminal distribution and count, then overall, without looking at the details of particular spines or terminals, you could say that there are no new synapses. Yeah, there's no synapse or synaptic change in the synaptic connections between plus plus versus say plus minus minus plus and so forth.
Okay, so one reason why I'm slowing you down on this one, I'm sorry again, is because I'm trying to compare this directly with the with I think one that you'll talk about later, this Choi et al. Paper 2018 or something that used an eGrasp technique, but it counted the number. It had the ability to count synapses. And I'm just wondering why they didn't do something similar here that just say, here's a count. This is the number of synapses from plus to plus versus synapses from plus to minus versus synapses from minus to plus, minus to minus on a subset of minuses that are chosen at random. Am I missing something? Why are they doing this distance metric?
That's a good question. Yeah. I don't know why they would do that.
I mean, I can see that this would counting distance is also okay. Anyway, I just wanted to bring it up because I'm trying to compare with other literature. So
that'd be nice if they used the same method, the same stats. Yeah. And yeah, I will mention Choi, but I'm only going to mention Choi in relation to, say, the time window, the interval.
Yes.
So if there are other things you wanted to mention about that, you can bring them up now if they relate to this besides the method of counting spines.
Actually, I'm just wondering if this metric might be in some way less biased than the counting method. In any case, if it didn't say somewhere in this paper that, hey, we're doing it this way because this is a principled local way of comparing connectivity, then
I didn't see that when I read it. No,
I'll assume it's just.
But I didn't read the supplemental material very carefully, so
okay,
it might have been in there.
Okay, thanks.
All right, moving on. This is the comparing proportions of multisynaptic Bhutans versus single synaptic Bhutans in CFC versus CS. Some terminals are connected to multiple spines, usually residing on different dendritic arbors. Multisynaptic Bhutans are believed to synchronize outputs to several downstream targets. At least that's what they state. I'm not sure that's the only hypothesis what they're for. Improving fidelity of neurotransmitter signaling within circuits. They hypothesize that the acquisition of new memories involves a shift in the equilibrium among conventional single synaptic Bhutans and multisynaptic Bhutans. And when they did the study, what they found is although multisynaptic boutons were present on both the minus and plus Schaefer collateral axons of fear-conditioned mice, their counts on plus axons were 1.6-fold higher. Their total spine-to-terminal ratios were elevated by 18%. And plus MSBs were coupled with 50% larger fractions of all innervated spines, the plus MSBs, than their minus counterparts, due to differences in both MSB abundance and their complexity. 26% of the plus MSBs contacted more than 2 spines, representing a 2.4-fold increase from the 11% in their minus controls. This increased the size and diversity of connectomes of the plus axons, and they supported this with quantification of separate dendritic branches receiving inputs from each terminal, fractions of compound synapses formed by Schaefer collateral fibers onto neighboring spines on the same dendrite, and average numbers of different dendrites whose spines are contacted by each multisynaptic bouton. So axonal networks are reorganized in the CA1SR through multisynaptic boutons in a stimulus-specific manner. But MSB target selection was not biased towards co-activated CA1 pyramidal neurons. The apparent recruitment of false-negative neurons that were not engaged in the immediate response to external cues is seemingly then at odds with the Hebbian model. Okay, they then went on to looking more carefully at spines and terminals for these Schaefer collateral, the plus Schaefer collaterals in CFC versus CS. They extrapolated synaptic weights by plotting in log normal distributions of spine head and nerve terminal volumes. Spines become enlarged during long-term potentiation and shrink during long-term depression. As per patch clamp, or in accordance with their patch clamp results, minus and plus CA1 pyramidal neurons of fear-conditioned mice had no detectable differences in spine head volumes, irrespective of axonal labeling. However, of 2 dispersed spine populations categorized solely by the activity history of contacting axons, the spines innervated by plus Schaefer collateral fibers were enlarged. And this is in figure 4C, which of course is too small to look at correctly right now, but you ought to be able to make that out. And it's supposedly statistically significant. The terminals of plus axons were also markedly bigger, and both of these results only apply to the CFC mice. The spines coupled to single synaptic boutons and multisynaptic boutons of the plus Schaefer collateral fibers were enlarged to similar degrees, with 1.4 and 1.3-fold increases in median head volumes, respectively, with smaller standard deviations than in the CS mice. Plus single synaptic boutons and plus multisynaptic boutons had the same 1.6-fold increase of median terminal volumes, despite the difference in bouton morphology. And only the plus MSBs had a much higher size variability than their minus counterparts. No changes in the median sizes of either synapse type occurred in plus pyramidal neurons activated by the context stimulus alone. They then calculate the Spearman rank correlations to define the relationships between extrapolated post and presynaptic weights in individual connections. They performed pairwise comparisons of volumetric measures for terminals, spines, postsynaptic dendrites, and axon-spine interfaces. Intuitively, a Spearman correlation between 2 variables will be high when observations have a similar rank between 2 variables and low when observations have a dissimilar rank between 2 variables. What they found is that the volumes of SSBs formed by minus axons strongly correlated with those of contacted spines. Correlations between the same parameters in minus MSBs were only moderate. These differences were even more pronounced among the plus SSBs and plus MSBs of previously activated CA3 pyramidal neurons. The sizes of presynaptic boutons and the number of neurotransmitter vesicles in each bouton strongly correlated with the number of innervated spines, and that explains, in a sense, the physiological relevance of changes in the plus MSB morphologies. They then subdivided connections of plus MSBs by the activity history of CA1 pyramidal neurons. Neither the distributions of spine head volumes on minus or plus dendrites nor the correlations between pre- and postsynaptic structures were significantly different, indicating that the tuning of postsynaptic weights is not restricted to co-activated pairs. Thus, the persistent restructuring of CA3-CA1 synapses after associative learning appears to be non-Hebbian at a cellular level. Memory encoding coincides with sustained augmentation of synaptic weights, as evidenced by shifts in the distributions of synapse sizes elicited by CFC but not CS. In line with analyses of neuronal wiring, these events are prompted by presynaptic mechanisms that do not require activation of postsynaptic neurons above threshold for FOS induction. The experience-dependent growth of plus MSB terminals is attributed to higher complexity of target interactions. And finally, they carried out a study of mitochondria and spine apparatus in CFC and CS. They reconstructed membrane organelles essential for energy metabolism, protein synthesis, and intracellular calcium signaling, the mitochondria and smooth endoplasmic reticulum, SER, the postsynaptic calcium homeostasis of glutamatergic pyramidal neurons, as well as local translation in spines, are thought to be regulated in part by a part of the SER network known as the spine apparatus. Prior EM studies had raised the possibility that subcellular localization of these organelles is influenced by sensory stimuli. And what they found is that mitochondria were generally more likely to be anchored in multisynaptic boutons than in single synaptic boutons. In CFC mice, more mitochondria-containing terminals tended to occur along the plus Schaefer collateral axons, although the difference wasn't statistically significant. But spines receiving inputs from plus Schaefer collateral axons of CA3 pyramidal neurons had a notable CFC-specific loss of spine apparatus, regardless of presynaptic bouton type. Median mitochondrial volumes were increased in terminals of plus Schaefer collateral axons by 1.3-fold. So presynaptic activity correlated with learning differentially affects, not activates, the organelles that maintain synaptic homeostasis and define the propensities for short and long-term plasticity by promoting the local function of axonal mitochondria and disabling the spine apparatus in spines. So now that we've gone through each of the experiments, we get to a summary. And they list out several core principles. Engram synaptic architecture induction is driven by presynaptic activity associated with specific sensory stimuli. Their expression transcends simultaneously activated neurons. They involve multisynaptic boutons. They then suggest that multisynaptic bouton remodeling underlies representational drift, and it's still not entirely clear to me that they made a solid case for that. I think that a lot of their more speculative discussion points, when you read them at the end there, are lacking what I would think is the longitudinal evidence. They argue that the Hebbian plasticity does not account for the wiring and sustained experience-dependent rewiring of the cellular substrates underpinning memory traces, and that this is made apparent by the higher resolution of their study. They propose a strategy whereby neurons that engage in memory encoding do so by diversifying the postsynaptic recipients of chemical signals from the same pools of neurotransmitter vesicles residing in each multisynaptic bouton, while maintaining the same numbers of individual nerve terminals and dendritic spines in a steady state. They speculated about a correlation with general intelligence by pointing out a higher density of multisynaptic bouton innervated spines in humans compared to mice, but that, again, seems really speculative to me. Now, here's some thoughts I had going through this, or considerations. One way to look at it, considering that there were morphological changes in SSBs, MSBs, and postsynaptic spine volumes together, even if the postsynaptic spine volumes weren't necessarily only the plus postsynaptic spine volumes, is that the study may be pointing to high-resolution detailing of morphology changes that mediate what is nonetheless Hebbian type of learning. At least that's one way that I feel one could interpret this. Another important factor is that in mice, retrieval of remote episodic memory is most dependent on an intact hippocampus within 24 hours of learning, and then the dependence diminishes over weeks and months, where a paper that Ken pointed out to me, the Taro et al. Letter in Nature from 2015 on impermanence of dendritic spines in live adult CA1 hippocampus notes CA1 spine turnover dynamics, first of all, differ sharply from those seen previously in neocortex, and that kinetic modeling of turnover of basal dendritic spines suggests a mean spine lifetime of 1 to 2 weeks, with a full turnover a little bit later than that. Also, noting the different results compared with the Chou et al. Results we had discussed in an earlier journal club, not a journal club this year, but earlier than that. I think that if I'm correct, the Chou data were obtained 2 days after contextual field learning. So this would be potentially in a significantly different domain than what we saw here. It's possible that at least in the CA3, CA1 circuit, initial involvement with episodic memory is, it's largely LTP dependent and dependent on spine changes that will already have receded to a great degree and not ultimately encoded by those structural changes. And going back to our terminology of caches, you could say that the cache is flushed for reuse. I think that the behavioral test they conducted after 7 days couldn't specifically test whether the same neurons in hippocampus were involved. So the behavioral outcome, whether that was being driven by the same neurons as the ones that they labeled, wouldn't have been clear. If we combine what this paper is demonstrating with our previous journal club on reactivating different cells during updating, then it begins to hint at a very temporary involvement of specific hippocampal patterns and fairly rapid repurposing. This is a question by Ken. Yeah, go ahead.
Yeah. On what you just said, the retesting, you were talking about in this paper, right?
Yep.
So the retesting, if you look, it was only done in cohorts. They made sure not to do it in the same mouse that they were doing this study on, which I think is absolutely crucial from your timing point of view as well, because if they had, if it had been consolidated and then they had tested it again, that testing would have brought it back into the hippocampus, maybe not with the same neurons, but
they wouldn't have ended up labeling those because they wouldn't have administered TMP again, right?
Absolutely. But I feel like, well, this is one of the things that I forgot to ask you at the beginning. I just want to make sure. So the contextual fear conditioning, it was a one-time deal, right? Yeah. It was one-time deal. No retrieval even for any of the mice at all. Okay.
Yeah.
I mean, hell, I would probably forget that I got back a week ago if it, if I never thought about it or something.
Yeah. Yeah, I think a difference between 2 days and 7 days, that might make a difference. Yeah. We can stay on this slide for a while if we want to discuss more the, the non-Hebbian outcomes and whether we believe them, whether we don't believe them. I have 2 more slides after this. One is trying to connect this back to memory decoding and seeing where it fits in, what it tells us, what it doesn't tell us. And then I have something completely separate, which is kind of an add-on question that came up to me while I was working through the paper. I think Ken was first.
All right. Well, I'm not sure if this is the right time, but what I would love is kind of your best explanation. Let's say this is, let's take the results at face value and say that this is presynaptic. I mean, there's presynaptic learning. It's no doubt. Let's say that this is just presynaptic learning. How would that, what's the model for that actually resulting in a memory that can be retrieved?
Yeah, this is why I kind of hinted at, you could look at this as something that still sort of fits within the Hebbian characteristic of
You have
learning.
to kind of like
If you have presynaptic changes that are definitely stimulus dependent, and then postsynaptically, you can have an elevated or an enlarged volume, say, in your postsynaptic spines, whether it's on the co-activated or non-co-activated ones, then this can allow you to still create memories where there is co-activation. Plus, you may be recruiting something else into that memory. So it's not necessarily a negative to the Hebbian learning, although it may be a bit of a washout. So I could see this working to a degree, and it might even help in a system where you're trying to recruit cells into temporary engrams of a sort for a moment. Maybe. But I'm not entirely convinced that this is everything that's going on, because again, I think that the 7 days versus 2 days difference and other questions we have about whether these particular mice actually learned what they were supposed to learn and still remembered it, I think those are bigger questions, just my personal take on it.
Okay. Just before Ariel goes, the Go-To et al. Paper as well made a pretty strong statement about if you erase hippocampal, if you erase consolidated memory or prevent consolidation of memory in the cortex, then the hippocampus is not a good retrieval after even a couple of days. I don't know how that squares with the Tardo 2015 result, but all of these results kind of hint that there's a very short window.
Right. Yep. Ariel.
Yeah, related to that. I realized I don't know why they picked 7 days after labeling. Like, I know for it to be a long-term memory, you've got to wait, let's say at least 24 hours, maybe 48 hours. But I assume the expression of the labeling peroxidase, whatever it is, probably doesn't necessarily take a full week. And yeah, does anyone have any idea why they picked 7 days versus 2 days versus 2 weeks? Like, they could have picked any time. Yeah, that's all.
Yeah, that's a really good question. I have a feeling they mentioned something about that, but it's not coming to me right now. So I'm going through the paper at the moment, but Ken or someone else, if you happen to remember.
I Control-F and clicked for days through the paper, and I couldn't find an obvious justification that way. But
well, I think it's a really good question of whether they had to do that from a techniques point of view, and I don't know if that's the case. In the paper, they make it seem like they're interested in remote. If you do a Control-F and look for the word remote memory, which Randall, I would love for you to define what remote means in this context.
Yeah, funny, huh? Because I think last time around we were pretty sure that remote meant already consolidated memory in a sense, right? But but here, what does it mean then? Yeah, let's see.
Because they're saying, oh, we're studying remote memories. We're interested in drift, and I can't look at this and think that they started with that study.
It's not clear that they're talking about consolidated when they say remote, but they do say that this is about a history, temporally remote history of correlated activity or correlated excitation during associative learning. So it doesn't necessarily say that a different mechanism's involved or a different region, just in time. Yeah. And I mean, that of course does correlate with having used 7 days, because then you've got, that's relatively remote, but.
And they make that clear, probably because the reviewer forced them to, in the discussion. I should actually look at where they say this. I had underlined it in the discussion. So they say in the discussion, they have limitations of our study. They say optical imaging of FOS-inducible transsynaptic markers suggests that co-activated CA3, CA1, same thing, preferentially connect with each other at earlier time points after associative learning. And they reference the Chou et al. So they are referencing the Chou et al. And then they say beyond the differences and pointing out that their results are contradictory with it. And then it says beyond the differences in imaging resolution and data analysis, discrepancy between our findings and prior work may reflect the possibility that non-Hebbian mechanisms, as described here, are preceded by an initial phase of Hebbian plasticity.
Right.
So they're essentially agreeing with everything that we're saying about the study in that one sentence in their limitations.
Okay. All good. Still, I'm kind of curious why they chose 7 days, because there must be a very specific reason, right? Because you don't just choose 7 days rather than 2 days, unless, say, for example, they were worried that the morphological changes they were looking for would not yet have happened or something like that. There has to be a really good reason to do that. Which I still haven't found.
Did you say you had a couple of slides left as well? I'm curious to see.
Yeah, I had a couple of slides left. I mean, they don't directly address this question about the non-Hebbian nature, but yeah, go ahead. Samuel?
Hello. Not really my area of expertise, but thank you for the summary so far. Just on the multisynaptic boutons, it seems from my reading that majority of the remodeling changes have to do with these boutons as opposed to the single synaptic ones. Is there, what is your intuition tell you why that is? And is there anything specific about these engrams that having multi-bouton ensembles is driving this remodeling?
I don't personally have a very strong intuition about it. Given that what we already pointed out about the limitations of the study, I'm almost ready to say, okay, there were probably a number of changes. Most of those changes have decayed or disappeared by now. The multisynaptic boutons had the most, were the most resistant against that kind of decay or, dissipation, and therefore they're still there. I'd be willing to just accept that as an answer. I don't know if Ken has another one, but see, I don't have a strong intuition as to why.
That's exactly how I would interpret it as well.
Wow, thank you.
Yeah. Okay, so this is what would have happened if they'd done the same exact study using EM and everything, but had done it 2 days later, 2 days instead of.
Hopefully, they're following up with a study. I mean, I would suspect that somebody has, said, "Hey, this is great," because that study with this study would actually tell us something about what the time changes.
Yeah, I mean the same.
Sorry, go ahead. Yeah.
The same for if, as you note, they've done it like for the equivalent of the motor cortex papers that we've looked at previously, where you see those like pretty notable differences in spine densities between the trained and untrained animals. Forget the time course there, but I think it was still there quite a few days later as well. So yeah, there's a lot of comparisons to be done depending on the time course.
Yeah, and interestingly, that kind of slides right into what my next slide is about. Because I think there are some things that we can at least take away from the study that are useful. I don't think the study is necessarily a strong contender for the prize, not just because of the limitations, but also even if everything were perfect, it would be difficult to use the methods shown to distinguish or identify one of several possible memory engrams. Although maybe in the methods area, they could be a contender. What this does do is it takes a closer look at precisely the kinds of data that you need to pick up on in specific brain regions when using static data to attempt to identify differences in connection strengths that make out representations relevant to engrams. And being a hippocampal study, it's probably not the most dominant type of information representing what we would consider markers of long-term memory or of engrams. But it's a reminder, again, that we need to think of memories and their representations not merely as a spatial collection, but also in terms of synchronization, which they brought up, timing, spatiotemporal. And it's a reminder that we need to study particularities of memory in multiple brain regions, because the same study done in neocortex would be another interesting thing to see. It's a reminder to consider different memory systems that apply over time, so transitions from one to another and on, because of all the different mechanisms involved. We've seen this already in other papers that were presented here, taking into account working memory and such things. It's a case study of a type of possible memory relevant detail in the data that one might have to pick up on that would not be caught, if what they're saying, if this multisynaptic versus single synaptic thing really matters, that you wouldn't pick up just by counting synapses, as they did, for example, in the recent Drosophila reconstruction papers. And it isn't even clear if what was identified here constitutes a memory engram, or maybe the ghost of a memory engram. But anyway, still, there are clearly morphological correlates here, data that you can get from a static snapshot. And it suggests that we do need to carry out more detailed simulations when we're doing simulations, containing many more pieces of information in order to subsequently tease out in these emulations or simulations what you have to focus on, what is your limited set of n parameters that are relevant to what we might call an engram, so that you can have something that you can then automatically detect in a static snapshot and make a correlate for. So I think there were, for me anyways, just because this was an EM study, it was interesting to see, oh yeah, so, had this been done 2 days later or something like that instead of 7 days, and you still did the same thing, and you could look at all these different contributors to the strength of a connection, and therefore to what makes up an engram, you could start to kind of tease out what matters and what doesn't matter. It was very interesting. Then I have one final slide here, and this goes in another direction, and it comes from a recent conversation, actually a conversation that I think Samuel started. And it came up again in my mind as I was looking through this paper, and I was thinking, oh, another hippocampal study, whereas hippocampus is not normally what we think of as, say, the place where long-term memory is stored. And another study of a different mechanism, and everything relates back, of course, to behavior, in this case of the contextual fear conditioning. So again, it's kind of a question we've had a few times. What is a memory engram really? How does a memory engram differ from stimulus response behavior? And would a full catalog of an organism's behavioral protocol be essentially the same as the content of its memory? And this relates to some important things about the consideration of whether or not details from the architecture, from a static snapshot like this, add EM resolution, if these are really important, or if physiological and architectural details are important at all, or if reproducing a behavioral map would be enough to satisfy the success criteria of an emulation. This is kind of like this tug of war between what used to be called lo-fi and hi-fi emulation. So yeah, it's really the question, what is a memory engram? What do we need to care about? Why the one, why not the other? And please chime in, because this has been on my mind.
So I think I understand the question if you're asking, would just having a list of behaviors that some particular organism does and being able to reproduce those behaviors in using an arbitrary architecture, would that count as successful reproduction of the memory behavior of that animal? What I don't think I understand is the question of exactly how that's related to what is a memory engram, insofar as a memory engram, I think isn't that the substrate of a particular memory that we're normally trying to identify? So it's you look at some animal and you can see that it's experienced one particular sort of experience versus another based on having this particular neurophysiological substrate versus another. Maybe
right.
They seem like two different questions to me.
Yeah. Maybe it's not stated quite right, because it's more the comparison between a memory engram and a known stimulus response behavior. So if you have a memory of something, like foot shock, I go in here, foot shock, and then I guess there's a behavior attached to that. You get out or run or whatever. That relates to a memory in a sense, right? So you've got the memory, you've got the behavior, the two are closely related. And then if you look at all the memory engrams and you look at all the behavior, you could say this is the same sort of thing. Now, I'm not saying it's the same thing. I really don't think it is. So I'm putting a straw man up here, basically. It's because of the conversation of this black box modeling. You take the whole thing as a black box. If you can only watch all the behavior long enough, then you capture everything that's going on in the system. Now you have its memory. So this solves the memory decoding challenge because you have watched all the input and output, you've captured all the memory. It's not from a static snapshot, not that, so it doesn't win that, but it would say solve the problem, say some people. And yeah, I guess I was looking for good arguments against that. The best arguments that I have is, number one, how do you know you've captured all of the behavior? Because there are so many variables inside the black box. How long do you have to watch? Number two, what if you care about the states of subsystems inside the black box? These are the sorts of things that we care about if we're thinking about, say, thoughts and internal experiences. But, a neuroscientist who's mostly interested in behavior might not. And so I was kind of surprised, actually, when Samuel told me that there is a very well-established neuroscientist who doesn't seem to see a difference between these two things. And you, Samuel, feel free to chime in because you know the conversation we're talking about here. It's having had that conversation and then going through this paper made me think, okay, how do you deal with that view of memory?
Mm-hmm.
Like, why is that not enough?
I think the second argument you mentioned was one I was trying to formulate how to articulate. It's kind of similar. I just revisited an earlier talk from Konrad Kording on this confounding problem of possibly multiple neurons synapting onto another neuron, and you don't know where the activity is coming from. Where you don't have to ask. You can treat the system as a black box model and not even look at the brain activity, and you can look at my texts, and you can assume based on that I would pass a Turing test. So this conversation that you're referring to, we talked about an established neuroscientist who said that if I can record correlationary data about the input and output and then some behavior, I will be able to create a version of me, of Samuel, who passes a Turing test. But I said, you don't even need the brain data for that. So, yeah. Just for shedding some context on the conversation that led here.
I mean, but I guess that's an open question that's why it's kind of different to the memory decoding challenge, I would have thought. Insofar as you can collect some arbitrary amount of behavioral data, use that to plug into your black box, and then hopefully generate something which can display the memory behavior going forwards in a way that you would predict. And that might work, or it might not work. I mean, I feel like I remember another talk of Conrad's where he talks about how you'd need to collect some enormous arbitrary amount of data to actually get that to work properly.
Mm-hmm.
But again, that's just different from actually understanding and being able to extract a memory or decode a memory or something. So I guess it's do you want? I guess I just see these as two different things. One's do you want, in a functionalist sense, to be able to just reproduce the system's behaviors, or do you want to actually understand how it's working?
Yeah, I'm asking the question because I'm looking at how to best clarify the difference. And the reason I feel that memory does come into it is because someone who thinks of the black box and having built something that works like the black box, that has the same stimulus response behavior, might feel ready to say, well, of course I've captured the memory that was inside of this system. If I didn't capture the memory of what was inside the system, how could it behave the same?
Well, I guess but you don't know to what degree. Like, if you captured it to 50%, 60%, 90%, 99%, like, if you produce a perfect emulation, then you've got a good argument for, like, that will arbitrarily going forwards, like, continue to play the behavior. But
so you're saying the best argument is the argument that you might need to collect way too much data, more data than you could possibly collect in the lifetime of the organism.
I don't know if it's the best, but it's definitely one that immediately occurs to me as a problem. Is like, how would you know and be able to verify that you'd actually got everything you needed?
All right. Yeah. Yeah, I'm happy to leave it at that if nobody else has any other insights. Yeah, this is just a question that popped into my mind while I was working through the paper.
So we have another hippocampus paper coming up in 2 weeks.
We do, don't we? Yeah.
I also, you mentioned behavioral time scale plasticity. I believe that was the term. Really have been thinking we should do a paper on that at some point, because I think that is a particularly relevant thing. If you have one, I mean, we could go back to the original. I think it was Mingi. But if there's one that's even better than that, and that you know of, send it over and look at it.
Yeah, sure. Yeah, that's interesting.
Cool. Thanks very much, Randall. That was fun.
Yeah, my pleasure.
Thanks, Randall. This is great.